Hi, my name is Eli Draizen, and I'm a PhD student in Phil Bourne's lab at the University of Virginia. I'll be talking today about deep learning of protein structural classes, any evidence for an Urfold. Most structural bioinformatics problems relate to sequence structure-function relationships and how different proteins have evolved from each other. One way to study protein structure is to use a hierarchical classification system because it provides an important framework for understanding sequence structure-function relationships. There are several different databases which perform hierarchical classification of protein structures, such as CAF, SCOP, and ECOD. For the purpose of this talk, we'll be focusing on the CAF database, where it relates to the class, architecture, topology, and homologous superfamilies for different domains in the protein data bank. Say you're given this protein and you want to figure out what its function is and if there's any other related proteins to it. The first thing you do is look at the secondary structure elements inside of it. The secondary structures can either be all alpha, mostly beta, or a mixture between alpha and beta. In this case, you can see that it's going to be mostly beta. The next thing you look at is the architecture, or the 3D arrangements of the different secondary structure elements. Here you can see the different beta strands form two different faces of a sandwich. So we call this architecture a sandwich. The third thing you look for is the topology, which is the 3D arrangement and pattern of connectivities between the secondary structure elements. Here you can see the directionalities of the beta strands and the connections between them. From this point, you can say it's going to be an immunoglobulin-like protein. The last thing you look for is the homologous superfamily. We try to find other proteins that are thought to have a common ancestor. This is usually found via sequence analysis, trying to usually greater than 20% sequence identity. This hierarchical classification system works really well for many proteins, except there's a few edge cases which makes it difficult to find related proteins. One such example is the protein that we've been studying in our labs called the small beta barrels, such as the SH3 domain or the OB domain. These proteins exhibit architectural similarity despite topological variability. You can see the structures are the same, but the connections between the secondary structures are different. And as you superimpose them, this becomes even clearer. You can see that the fifth beta strands overlap with each other, 1 and 4 overlap with each other, 2 and 1, 3 and 2, 4 and 3. So while the secondary structures are different in the overlapping, but the overall structure is the same. Another way to look at this is using the primary sequence along with the secondary structure and tertiary structures of these. You can see that the beta strands are going to be in different orientations and different positions in the sequence. So if you align them, you're just not going to be able to find a direct match. One thing that's so interesting about these small beta barrels is that they're very robust. Any changes to the sequence, even vast changes, can result in the same structure. Another useful, interesting thing about these small beta barrels is that they form different oligomers. They're also used as protein scaffolds in a multi-domain protein. Because of this, we think that there might be a new entity between architecture and topology, which exhibits the 3D architectural similarity despite topological variability. We propose to call this new entity an Urfold. From manual searches in the protein data bank, we found a few more examples, such as the P-loop and TPases, where you can see it has the same arrangement of secondary structures, but the connections between them are different. Same with the KH domains. The beta strands are in the same orientation, but the connections are different. Last, we also found some proteins that are found in archaea and sometimes that are bound to, or known for tata binding. But the theme remains clear, that these structures are very similar, but the arrangement of these secondary structures are different. So is there a new way to classify these to make sure that we can understand these relationships a little clearer? So if we look at the current view of clustering, we can see that the SH3 is going to be under the architecture of rolls, and the OB is going to be the architecture of barrels. However, these are very similar, and we think that they should be clustered in the same way. We're also showing the immunoglobulins in this tree to kind of show it's like an outgroup, showing that it has a somewhat different evolutionary history, even though it has several beta strands and is it might perform similar functions as in binding, but for the purpose of this, it's going to be an outgroup has a different evolutionary history. However, what if we were to cluster it in a different way to show that there's similarity between SH3 and LB in the case of a small beta barrel? So if we put a new hierarchy or a new entity between architecture and topology, it might bridge these two in a new way. Another reason why we think there might be a new entity is because there's a discrepancy between the counts of each node in each different entity. So a jump from 41 in the architecture to 1391 in topology, and 1391 to around 6,000. We think if there's a new entity somewhere in between here, there might be a more even distribution of counts. The main purpose of this talk is to describe a new algorithm we're developing called Deep Urfold, which is trying to learn local substructures of biophysical properties and geometry to bridge these gaps in hierarchical classification systems. So one way to do this is to try to create a new similarity metric between protein domains, to try to reconstruct this new dendrogram. We do this by creating different autoencoder models for each different homologous superfamily, where we input one protein domain using all the representatives from CAF. We encode them using a three-dimensional convolutional neural net, which is trying to find an efficient compression algorithm to try to decrease the volume while increasing the number of features until we get to a latent space or a bottleneck. And then we try to decode them or reconstruct them using the three-dimensional CNN and try to reconstruct what the superfamily would look like in terms of this model. So in order to do this, first we train one model for each different superfamily using the representatives from CAF. And then we subject to the different superfamily representatives to all the other superfamily models. Finally, we can create a new hierarchical clustering with these neural network scores, such as the mean squared error or the area under the curve. And hopefully we can recreate a dendrogram similar to this. However, you can't directly put a protein structure into 3D CNN. It has to look like an image first. So we do this by voxelating all the atoms. This is done by centering the protein in a 256 angstrom volume, and then trying to find van der Waals spheres around each atom and discretize them to fit 1 angstrom voxels. You can see that the volume around this protein is going to be sparse. There's no atoms occupying the rest of the 256 angstrom volume, so we don't need to annotate it. Using sparse CNNs allows us to train many more proteins at once with a large number of features without the computational runtime that would be required if we use all the dense features. As you can also see in this figure, there's going to be different feature sets for each voxel, which are three-dimensional pixels. And then each atom, or each voxel within one atom retains this, each voxel within one atom retains the same set of features. We also treat covalent bonding, where we find overlap between atoms, or voxels that are shared by one or two atoms. And we do this by maximizing the features. Next, we calculate different atom-based biophysical chemical features for each atom, such as the atom type, residue type, secondary structure, accessibility, is hydrophobic, is positively charged, or is electronegative. All of these are going to be Boolean values, so 0, 1, yes or no, except atom type and residue type, which are going to be categorical features, which are going to be just grouped of Boolean features. So is oxygen, is nitrogen, or is alanine, or is cysteine. So once we have that, we can train these different autoencoders for each superfamily. As you can see, these models converge around 30 epochs using a learning rate of 0.1 and a batch size of 16, and using the atom optimizer. So during each iteration, we rotate the structures randomly, so we can get rotation invariance of these different models. Once we do this, we can also try to do feature selection, where we calculate the AUC for each feature in each voxel and we average between all samples. We can see that secondary structure is very important. Is the atom buried? The atom type is electronegative and is hydrophobic. However, what we found pretty interesting is that the residue type is not as important. This might be indicative that the structure is actually very important, and using atom-based features might be more informative than just residue features. One test case to see if our models are working is try to do feature construction of one of the most important features, which is electrostatic potential. Here you can see one immunoglobulin domain, and we try to reconstruct the electrostatic potential. So we encoded the electrostatic potential is less than 0 as 1, and electrostatic potential is less than 0 or equal to 0 as 0. As you can see, it tries to reconstruct the electrostatic potential. It does it quite well with a mean square error of 0.5, but there's definitely room, it's definitely not perfect in the sense that it's also training and learning from every different calf immunoglobulin domain. So that might influence what is reconstructed in terms of this model. Finally, we can use these models to try to reconstruct or try to find new distances or similarities between the different superfamily models. On the left, you see a confusion matrix where we apply the structures from one superfamily into all the other superfamily models, where we calculate the AUC and the mean square error. On the right, we have a dendrogram, except this is all going to be taken, all the samples are going to be different. On the left is going to be average for all the different structures in that one family, in that one model, but on the left is all expanded out. And on the right, it's expanded out. One thing we noticed from this dendrogram is that it's not split cleanly. So there's a lot of cross-contamination between the different superfamilies. So we think that because it's clustering based on biophysical properties as well as geometry, that there's going to be some similar biophysical properties and geometry that are shared between all these superfamilies. But there's not as much mixing between the OP fold and the SH3 fold. So because of this discrepancy and because it's not a clean split, we think that there must be some similarity between these structures. And if we could find these local substructures, we might be able to identify what an Urfold is. However, this is just a proof of concept. We only ran it for 3 immunoglobulin domains. So we plan to run it through all of the calf superfamilies. We also plan to identify what are the local regions of importance by using layer-wise relevance propagation, where we can go through the neural network and try to identify the scores that, or the voxels that led to positive scores in these networks to try to figure out these different regions that are important. So in conclusion, we think that there may be a potential new entity called the Urfold that exists between architecture and topology to represent the 3D architectural similarity despite topological variability, which we showed by examples between SH3 and OB domains. Next, we developed a method to find relationships between superfamilies using 3D CNNs. And finally, we showed that 3D CNNs can learn discriminatory biophysical properties and geometries for different superfamilies. In the future, we hope to use this framework to scan the entire protein universe to find more Urfolds. I would like to thank everyone in my lab for support on this, especially Phil, Cam, and Stella for devising this project and coming up with the Urfold idea. I'd also like to thank Manuka, Saad, and Kwan, who were Masters in Data Science students at the University of Virginia, who worked on this project as part of their capstone. I'd also like to thank funding from the University of Virginia's Fellowship in Data Science program, and the Presidential Fellows. Thank you. If you have any questions, please come to the question and answer portion, which is going to be Wednesday, July 15th at 10:40 a.m. Eastern Time. Otherwise, you can contact us through many different methods. Thanks.